Welcome, everyone. Hello. Welcome to the Global IMMUNOTALK. My name is Matteo Iannacone, and it is a great pleasure to introduce Michael Sixt as today's Global Immuno speaker. Michael is originally from a small town in Bavaria, Germany, called Weiden, and he went to medical school in Erlangen, Germany, where he also underwent clinical training in dermatology. He then did a postdoc in Lund, Sweden, and was then appointed principal investigator at the Max Planck Institute of Biochemistry in Germany. And in 2010, he was recruited to IST in Austria, where he is currently a tenured professor and also vice president. In my opinion, Michael is one of the most creative scientists I know. His lab studies the cell biological and biophysical foundations of cell migration, and he mostly uses leukocytes as a model system, and he employs very creative reductionist approaches to tackle fundamental biological questions. And amongst his most prominent discoveries, there's the notion that functional integrins do not contribute to migration in three-dimensional environments. He established the concept of haptotaxis, that is the directed migration along immobilized gradients in tissues. He identified polysialation as a critical post-translational modification that controls dendritic cell trafficking by regulating chemokine recognition. He showed that lamellipodial actin network of migrating cells undergoes geometrical adaptation in response to mechanical load. He found that nuclear positioning facilitates amoeboid migration along the path of least resistance. And finally, he discovered that leukocytes can not only migrate under low adhesion, but they can also transmit forces in the complete absence of transmembrane force coupling by using the topographical features of the substrate to propel themselves. And so in recognition of his work, Michael has received numerous awards, including the Young Investigator Medal from European Biophysical Societies, the Lieben Prize, an endowed professorship from the Peter Hans of Schneider Foundation. He is an elected member of the Young Academy of the Austrian Academy of Sciences and of EMBO, and he was able to obtain a consolidator and now a synergy grant from the European Research Council, which is the most prestigious European funding agency. So, Michael, thank you very much for having accepted our invitation, and I very much look forward to your talk entitled Leukocyte Locomotion, the Single Cell Perspective. Now, before we start, it is a tradition of Global IMMUNOTALKs to ask speakers a question to get to know them a little better, or perhaps to inspire the next generation of scientists. And the question we have for you is, could you share one of the most impactful decisions in your scientific career?
Okay, thank you. Thank you very much, Matteo, for a very kind introduction and for the invitation to this fantastic series. So I always watch the IMMUNOTALKs because, and I think it's really one of the few positive things that we got from the pandemic. So thank you very much. So you're asking me about the most impactful decision, and for me, this was clearly to leave clinical medicine behind and really turn towards full-time basic research. And it's actually not that I disliked work as a doctor, but I just really enjoy finding out how things work. And so I guess what I want to say is that it's always worth to try a few different things. And once you find something you're really passionate about and get the opportunity, then I think it's worth doing it. I never regretted it to really do full-time basic research. So.
Thanks, Michael, and I couldn't agree more because I made the same decision, so that resonates with me a lot. So thanks for sharing this. Now you can go ahead and share your presentation.
Okay, yeah. What I want to do is I really want to talk about leukocyte migration, leukocyte locomotion from the perspective of the single cell. So if you watch this Global IMMUNOTALK series, then I think there's almost no talk where it wasn't mentioned that cells migrate from one spot to the other. But we in our lab, we turn a bit around the question, and actually we rather use the immune system as a model system to understand how the single cell works. So I want to really take you along the journey how the inner workings of the cell make it possible for leukocytes to be very, very efficient migrators. And I don't have to tell you that leukocytes are the most motile cells in the body. But I still show you this movie, and I'm sure you have seen several of these types of movies. So this is a 2-photon movie that Mirek in the lab, together with Jens Stein's lab, did of T cells migrating in the lymph node of a mouse. So in magenta, you can see the T cells. In green, you can see the stroma cells, the stromal backbone. And we have the time running here. So within 20 minutes, you can see that the cells are covering an enormous distance. So they migrate around 1 cell diameter per minute, whereas the stromal cells are actually rather static. And of course, this is absolutely essential for the priming of adaptive immune responses and for the pathfinders, for the search for the right antigen presenting cells. And we once made a calculation that we found quite fun. So we estimated what's the cumulative distance that all the T cells or all the lymphocytes in a human body migrate within a certain time frame. And it's really a staggering number. So within 15 minutes, your lymphocytes migrate around 25,000 miles. So it means at the end of the talk, after around an hour, your hypothetical lymphocyte will have it made like 4 times around the equator. So this is clear that leukocytes are actually very, very motile cells. So if you talk about cell motility, then the central molecule we are talking about is really actin. It's and this is a G-actin monomer here. And actin is the most abundant protein in animal cells, which also makes it one of the most abundant proteins on the planet. So whenever we eat something, we taste actin. And this here is a movie of an endothelial cell that has actin labeled. And if you watch this type of movie, you get a feeling that really actin determines cell shape. Whenever you have a burst of polymerization, like in example here in the periphery, you also bulge out the membrane. And if you poison actin with drugs like latrunculin, so if you depolymerize it completely, most animal cells take a completely spherical shape. So all cell shape is determined by actin. And maybe something I want to mention at the beginning of the talk here, I won't go into too much detail on specific project, rather give a high level overview. And I will also not present a linear story. So I will mention the people who did the experiments here along the way. So this is how actin looks in the textbook. So the G-actin monomers assemble into F-actin filaments. The filaments have a plus end and a minus end. And at the plus end, the new monomers come in and can generate a force that bulges out the membrane. So this can push the membrane forward. But at the same time, like every force produces a counterforce, the filament pushes itself back into the cell while it grows. And within the cell, it's actually disassembled by factors. And it's again disassembled into G-actin monomers. This diffuse around in the cell and can then be again incorporated at the front. So this means this whole process of actin polymerization doesn't involve new protein synthesis. It's basically a recycling process where the actin can treadmill along and be the monomers are recycled to the front. So this is how it looks like in an animation. So you see here the F-actin filament growing at the tip. With every new actin monomer dropping in, the filament grows around 3 nanometer. And at the back, it disassembles and then the monomers diffuse. The assembly-disassembly process is dependent on ATP, so it costs energy. And every monomer that is added costs 1 ATP, basically. So this is how actin converts chemical energy into mechanical forces. So what Jan Müller in the lab did here is electron tomography of the leading edge of a cell here. So it zooms now through the set, and all the stripes here that you can see are actually actin filaments. So all the spaghettis are actin filaments. We can trace them and quantify them, look at their geometry. And what you can primarily see here is that really the leading edge of a cell is simply full of actin. And from this type of data, we can also calculate now how many filaments are actually pushing out at the membrane, at the front of the cell. And from this, we could actually make another calculation that we found fun, because now we know that every monomer advances the cell by, let's say, 3 nanometer. Every monomer costs 1 ATP. Now we can say, okay, how much energy does it cost a cell to move when it has the energy here of one banana, which is 400 kilojoule. So how far does the cell get with 1 banana? And the answer was actually, for me, totally unintuitive, because it's an enormous distance. This is actually, the cell can move roughly from the Earth to the sun. So one single cell just walking there with the energy of 1 banana. So this calculation is just an estimation, might be wrong by a factor of 10, but still it's a few million miles that the cell can move. Okay, so we already quite a time ago, when we were in Munich, we collaborated with Roland Willich-Söldner's group, and we employed a little peptide derived from yeast where we can actually tag actin. So we can couple it to GFP so that we can visualize actin. We call it LiveAct. And here you can see a movie of this is a fish keratocyte now, moving on a surface, and you can see the flow of actin. And I guess you can appreciate here that there's a constant stream of density variations from the front to the back of the cell while the cell is moving forward. So you already get a feeling that there's really assembly at the leading edge, disassembly at the trailing edge, and in between flow. But you can really appreciate the flow of the network and the molecules when we did here this laser cutting experiment. So we take a laser, cut into the actin cortex, and you can see that while the cell is moving forward, the destroyed area actually stays put in relation to the substrate while the cell moves forward. So there's really, the actin is really rolled out like a carpet while the cell is moving forward. We can do the same thing with a lymphocyte now. And this is an experiment Henri Versat did. So she put a T cell between 2 parallel surfaces, and these surfaces are passivated, so they cannot bind protein, so they're inert. And now we take lymphocytes, put them between the surfaces, and look at their actin polymerization. And you can see here, the lymphocyte polarizes. It points towards the upper left, so you can really get a feeling, okay, it wants to move there, but it doesn't advance. And if you look at the actin, you see it's constantly flowing backward. So this is because the cell, they have their actin treadmilling process running, but it can't couple to the surface because the surface is inert. So it's like a car driving on ice where the wheels are constantly spinning. So if we now coat the 2 surfaces with serum or even a specific adhesive ligand for the lymphocytes, then the cell can generate traction and turn into a moving object. And this you can see here. So now we have the cell that roughly has the same shape, it's polarized, but now it turns the actin retrograde flow into forward locomotion. And this happens because now the retrograde actin flow can couple through the membrane to the substrate and turn the retrograde flow into traction forces. And this is how cell migration looks in the textbook then. So a cell protrudes, it adheres to a surface, and then it turns the retrograde actin flow into traction. So the integrins couple through the membrane, outside to the substrate, inside to the actin, and there's not only one integrin, but in animals there are 24 different integrin heterodimers. Many of them are expressed on leukocytes. And you can now imagine that a cell has a certain equipment of integrins. Let's say the yellow cell has a set of integrin that can bind to the yellow surface. The red cell has another set that can bind to the red surface. So this causes sorting of the cell and compartmentalization in tissues. And this is one major principle how metazoan organisms are really compartmentalized. And there's even a fail-safe mechanism, and this is that the integrins, they do not only mediate traction forces and adhesion, but they can also mediate survival. So that means when usual tissue cells, when they lose their adhesiveness, they also lose a survival signal, and then they undergo apoptosis. And it's a special type of apoptosis that is called anoikis. So that's a Greek word for homelessness. So the cells fall out of their niche, they become homeless, and this is why they undergo suicide in order to prevent that they grow in the wrong compartment. And this is, of course, what happens in cancer cells. So anoikis is bypassed, and the cells can grow in the wrong compartment and create the mess that we know. So this is how mesenchymal cells move. So this is, they adhere at the front of the cell to the substrate and pull forward via adhesion. And this is what you can see here in this movie. So you have a fibroblast here, leading edge of a fibroblast in a collagen gel, and it moves downwards. And while it moves downwards, it pulls on the substrate, and you can see how the collagen fibers are pulled towards the cell. That's a typical mesenchymal type of movement. But if you now look at hematopoietic cells, then the situation is really different, because hematopoietic cells are notoriously homeless. So they are constantly traveling around, and they traffic transiently through static compartments, and they can traffic through very different compartments. So they have to be very promiscuous in their interaction with the environment. And accordingly, they use quite a different mode of locomotion. So I talked about the mesenchymal type of locomotion and showed you the movie here, but the leukocytes are typically described as amoeboid. So amoeboid primarily means that they are way, way faster. So they're about 100 times faster. And while they move, they actually don't deform the environment much. At least they don't pull a lot on the fibers around them. And at the same time, they're constantly deforming their body while moving forward. So accordingly, so you don't see the big traction forces. We found, and also others actually quite a long time ago, that when we delete all integrins in leukocytes, and that's the same for dendritic cells here moving towards the lymph node, or lymphocytes moving within the lymph node, the same for neutrophil granulocytes. So when we delete all integrins, the cells can actually migrate very well within the interstitium. So they are not totally reliant on integrins like the other mesenchymal cells. So this doesn't mean that they can do well in the organism, because what they can't do without integrins is, of course, extravasation. So I think most of you know the principle of the extravasation cascade, where the cell first rolls along the endothelium and then tightly binds to it and then transmigrates. So this paradigm involves a step where the leukocytes tightly adhere via integrins to the endothelium, and this holds true also. If you knock out the integrins, they don't make it through the endothelium, but once they're in the interstitium, they can migrate without the integrins. So we were asking the question, actually, what is the difference between a 2D surface or between a culture dish and interstitium in the mouse? And I already said it. So the prime difference is actually the geometry. So in the lumen of the blood vessel, the surface is two-dimensional, but once the cells are in the interstitium, they're usually surrounded by a fibrillar environment. And so they're really confined because the fibers push on them. And also, if they're in a tightly packed cellular environment like the lymph node, they're also constantly surrounded by things. So they're always in a 3D environment. And it turned out in the last years then that actually the adhesion dependency of cells in general decreases. So it's a by and large rule, decreases with the complexity of the extracellular environment. So on 2D, they are totally dependent on adhesion. In a fibrillar environment, they can move without adhesion. So question was, how does this actually work mechanistically? So here I show you again the experiment that André Versat did in the lab. So two parallel surfaces, lymphocyte slightly squeezed between, and then knocked out taline in the lymphocytes. So taline is a force adapter molecule that binds to the integrin. It's absolutely essential for the functionality of the integrin. So if you knock it out, you get dysfunctional integrins. But you can also do the experiment where you simply knock out the integrins, or as I told you before, where you passivate the surfaces so that the integrins cannot bind. So in any case, the same result. So control cells are nicely migrating between the two surfaces. The taline knockout cells, the integrin deficient cells, they can't because they run on the spot, because they constantly slip. But now Anne took exactly the same cells with dysfunctional integrins, put them into a 3-dimensional collagen gel, and the cells migrated totally normal. So now, of course, one could say maybe there is simply another adhesion receptor that bypasses the integrins and binds to the collagen, but we were following another thought, and that was based on this previous considerations that the more complex the environment, the geometry of the environment, the better they migrate without integrins. So we started to create intermediates between the two parallel surfaces and the fibrillar complex gel, where we basically incorporate texture into the into these two surfaces. So the cells have the same chemistry they are facing, but they have another geometry. And this is one type of example here, the experiment where Anne generated this channel that has riffles here at the beginning. That's the cell sitting in the channel. So it has riffles in the beginning, and then there's a smooth part. And if you take a wild type cell, a control cell now, you can see it's nicely moving through. So it really doesn't care if the part is riffled or if it's smooth. So it just passes through. But now if you take cells that have no integrins, or if you passivate the channel, then the cells also nicely make it through the riffled parts. But once they hit the smooth part, they can't move anymore, because then it gets slippery and they can't generate traction. But obviously in the textured part, they can do it. So we have a situation here that can be described in a very simple physical model. So you have a cell sitting between two surfaces. The cell is polarized. So there's actin flow from the front to the back. So polarity can either come from within, that they spontaneously break symmetry, or there can be an external guidance cue like a chemokine that steers the polarity in a direction. But whatever the source of polarity, you have the actin flow from the front to the back. And only if there are integrins now coupling to the surface, then the cell can move forward. If you don't have integrins, the cell is running on the spot. But now if you introduce a texture into the environment, then the actin flows along the curvature of the substrate. And whenever it meets the downslope, it can actually push with normal forces against the substrate and create a force that drives the cell forward. So this is a model that Raphaël Vautelier and Mathieu Pierre and Andrew Cullen-Jones proposed here with us. So for this model to work, of course, you need the period of the topography to have certain features. If the gaps are too big, then of course the actin flow can't properly meet it and generate the forward forces. So we tested this and Anne made here channels that have a texture with increasing periodicity. She took again the integrin deficient cells, put them in, a wild type cell would effortlessly move through. But now if you can see this integrin deficient cells, it gets more and more problems the more it gets up the channel, actually. And now it has to stretch and stretch in order to get into the next compartment. And only when it hit the next compartment, it can push forward using the topography. And now that's the end of it, because there's only a smooth part of the channel, and that goes on forever. So the cell is desperately trying to get in this direction, but it's slipping and slipping, and cannot gain any traction, and cannot pass through the channel. Also, there's somebody trying to help up here. So this is all, as I said before, driven again by actin polymerization. So here you can see a cell in a channel with these bulges here, and in white field, and in here the actin movie. And you can see at the beginning it's slipping, it's turning around, it manages to get into the next bubble, but now the period of the texture is getting smaller and smaller. So the cell is doing better and better. And here it's like almost moving with maximum efficiency up. And once it leaves the channel, it starts slipping again, because there are only two parallel surfaces and can't advance. So again, at the beginning, it has some problems, it's slipping. And now there's traction via the coupling to the topography. So this is what we can now call really, we think, the true amoeboid mode of locomotion, because amoeba is actually derived from the Greek word for change. Amoeba is the animal that changes shape, and that's something you can also see here. This is a amoeba that Ingrid and I fished out of our institute pond, and so it's an amoeba moving here and constantly changing its shape, and you can see morphologically it's hardly actually distinguishable from a T lymphocyte here. So they really use the very same principles to move forward. So if we now look at this model of movement, this is extremely simplistic, of course. So it's just involving actin flow, and the cell actually in this case doesn't even respond to the environment. So once you put it into this texture channel, it has to move forward. So, but reality looks, of course, much more complex, and in reality the cells do respond to the environment, and this you can see here in a movie by Patricia. So you have a dendritic cell here in a three-dimensional collagen gel. You see in gold the collagen fibers, and you can see that this cell is performing an enormous difficult task. It constantly has to make decisions, dive through the right pore, explore another one, find the bigger one, and make a lot of decisions, and also has to deform the fibers a bit, and I will come to that. So the cell actively interferes with its environment. And here I want to come to a second feature of amoeboid cells. So first one was they can move very well without adhesion receptors. The second is they actually move without proteolytically digesting their environment. So a mesenchymal cell, when it moves forward, it really digests the environment. So it digests a whole while it's moving forward. The leukocytes don't do it, and it's pretty clear that they couldn't even do it. They don't have enough time for it. They are too fast. And if you think of your leukocytes traveling 25,000 miles in 15 minutes, then you would be totally perforated in the evening if they would constantly proteolytically digest holes. So they don't do that. So they rather move along the path of least resistance when they move forward. And this is a principle that we tested here. So this was done by Jörg Renkewitz in the lab. So here Jörg put dendritic cells in a three-dimensional collagen gel and exposed them to a gradient that attracts them. So it's the chemokine CCL19 that attracts them. And now the collagen gel has two different densities. So here's a blob of a gel that has higher density. So if you look in reflection microscopy, you see it's high density here and it's lower density down here. And now all the cells, if I start the movie, they are attracted up. And you can see when they meet this very dense blob heads on, they can actually move through it, but they get very slow. But what they do is when they tangentially meet the blob, they actually move around it. And probably you can see it better in the tracks here. So these are the tracks color-coded for velocity. You see when they meet it head-on, they slow down but still move through the dense blob. But when they go tangentially, they decide to go around it. So they make a decision, and they choose the path of least resistance and choose rather a longer path length in expense because they can be faster there. So we again modeled this process with a more reductionist setup. So we put the cells again between two parallel surfaces, but now we built little pillars between the surfaces. And you can now see leukocyte is like going slalom between these pillars. And we can now arbitrarily shape the position of the pillars. And now here we have areas where the cells have very dense pillars and here not so dense pillars. Again, the dendritic cells here are attracted by a gradient of CCl19 going from bottom to top. And you can see that the cells actually prefer to go around the dense areas. So they rather go in where the pillar spacing is higher, but once they hit the dense areas directly, they can move through it. They are just slower then. But again, here they choose the path of least resistance. And this brings me to another defining feature of amoeboid versus mesenchymal cells. So in a way, amoeboid cells have something like an inverted polarity. They very much prefer to migrate with their nucleus at the front and the organelles, including the microtubule organizing center behind the nucleus. Whereas the mesenchymal cells, they have the nucleus rather at the back and the MTOC and the organelles at the front. So they move the other way around. And Jörg showed that they can use this, that leukocytes use this forward positioning of the nucleus actually as a sensing system to decide for the path of least resistance. So here I show you an example movie of a dendritic cell moving again through a forest of pillars, and they have differential spacing here. So here in orange are the bigger pores, and the smaller pores are in green. And now when I start the movie, you can see the nucleus of the cell kind of scanning the pores and always deciding, or in most cases, deciding for the bigger pores. So the cells use it like a gauge to measure the spacing of the pillars. And they know whenever the nucleus fits through, because this is the most bulky compartment of the cell, the rest of the cell will also fit through. And they take basically the nucleus as a lead here. So we found that very remarkable that in general that cells can really do these trade-offs between like a gradient and the density of the environment. And it just tells us that really cells are very competent and smart agents. They don't have to be micromanaged by the tissue. It's really enough if they get a high-level information, like the growth gradient goes in this direction, and then they're making their decisions locally and deal the best way with it and choose the best path. So, but now in the last part of the talk, I want to come to the point that how are these cells actually dealing with the tissue if they really want to go through a dense part? If there's a very steep gradient, they really want to go through a dense part, they do it. And if they have no alternative, they switch modes of motility here once they enter this dense part. And we know that leukocytes can even do something very extreme. They can even switch on the degradative mode. So these are experiments Christine Moussion did in the lab. So she put dendritic cells on basement membranes, actually. And basement membranes are totally tight sheets, so they only have a few nanometer diameter pores. So if the cell wants to get through, it has to digest a hole into it. And if dendritic cells are exposed to a gradient and sitting on this basement membrane for quite some time, they're able to digest holes into it. But this is something we don't know much about. So we don't know how they're actually switching these modes. What we know a little bit more, and I want to tell you about that, is how they deal with dense environments that they still can transiently deform. So it's about cellular mechanosensing. And you can actually see here in this movie of the dendritic cell moving through the collagen gel, where you can see on the right-hand side here only the collagen gel, that the cells do transiently deform the fibers. So they rattle a bit on them while they move through, but then everything more or less goes to the same position. So they don't digest the whole, but they transiently dilate it so that the cell fits through. So how can they do this? And it is actually, if you look into the known principles, how cells are mechano-responsive, then this is all based on the pulling principle, actually. That's all based on adhesion to a substrate. So the cell adheres, and then it pulls via actin on the adhesions, and even via actomyosin contraction, and then thereby it can sense the mechanics of the environment. But how does it work now in a leukocyte? Because if you want to pull on something, you need a handle to pull. So you need the adhesion receptor. But if the cell is either in a very soft environment where you cannot pull a lot because it's giving way, or if the cell doesn't have adhesion receptors that bind tightly, then they have to use another principle. So possibly they do it everything by pushing. And this is something we were exploring when we looked at the function of the Wiskott-Aldrich syndrome protein. So I want to come now here back in order to address this pushing function. I want to come back to the scheme I showed you in the beginning of the actin treadmilling here. So actin is polymerizing at the front, pushing out the membrane. But it's not only elongating, actin is not only elongating at the plus end, but there's also constantly generation of new filaments. So new filaments are nucleated on demand, and this happens mainly, so one major protein that does this, especially at the leading edge of a cell, is the ARP2/3 complex. So the ARP2/3 complex binds to the side of a filament and nucleates a new filament that branches off in a 70-degree angle. So it makes two filaments out of one. So the ARP2/3 complex is again regulated upstream via the wave complex. And the wave complex is activated via the RUC GTPase. And the second input that is activating ARP2/3 is actually a protein that immunologists usually know quite well. That's WASP, Wiskott-Aldrich syndrome protein, which again is activated upstream via the GTPase CDC42. So we look here in this movie now at the ARP2/3 complex in a migrating dendritic cell, and we can see there are two populations. So on the one hand, we have the one at the front, where you would expect it to nucleate the filaments that push out the leading edge. But then we also have a second population of ARP2/3, and these are these small dots here that are constantly traveling backwards and accumulating here in the cell body. And we found that these are actually, these two different localizations are separately activated via wave versus WASP. So the wave complex, as it was actually shown in other cells, is present here at the leading front of the lamellipodium. So it generates the filaments that drive the membrane horizontally forward. But if we look where WASP localizes, we found it in these spots here. So comes in little spots that travel backwards. And when we look now at the actin signal in these cells, then we can see on the one hand, the lamellipodium is pushed out here, round and flat, but at the same time, we have the spikes of actin here. So spikes and dots of actin. And these actin spikes, they coincide with the position of the WASP molecule, while the wave is here outlining the leading front of the cell. So what Florian Gettner in the lab then did is actually he probed cells migrating in deformable environments. So what he did here is he put dendritic cells under a pad of agarose. So they're nicely moving along a chemokine gradient between a cover slip and a pad of agarose. And the agarose, we can now tune in stiffness. So you can basically measure the stiffness via atomic force microscope of the cell and of the agarose. And you can have agarose that has the same stiffness as the cell, or softer or stiffer. And when the cell moves under this agarose, they're like almost flat like a pancake, like you can see here in scanning EM. And this is a transmission EM from the side. You can see that the cells are extremely flat. But when we look now in more detail, Flo saw that there are actually little bumps reaching up into the agarose. So, and these bumps contain actin. Don't show you that, but there's actin in, and it looks like they protrude into the agarose. And when you look at these bumps, when they co-localize with the nucleus, when there's a nucleus underneath, you can actually see that they do not only push up into the agarose, but that also pushes down onto the nucleus. So it's really in line with the idea that you have the wasp spots here that polymerize actin, then they push up into the agarose, and at the same time they push down into the nucleus, deform both, and thereby lift up the agarose. So if you knock out Wiskott-Aldrich syndrome proteins, or if you take wasp out, knock out dendritic cells here, these dots, these actin dots are completely gone. If you knock out the wave complex here by knocking out one component, which is called HAM1, then the dots are still there, might even be a bit more pronounced, but the leading edge is not round anymore because the cells don't have up to 3 at the front, so they just make spiky leading edges, a few filopodia. But important thing is here, the wasp dots are gone. And accordingly, if you look from the side on the cells, they're completely smooth on the surface, so there are no bumps reaching up into the agarose anymore. If now Flo did the migration assays where he looked at cells moving under very soft agarose, then there was actually hardly any difference between wild-type cells and wasp knockout cells. But if you tune the agarose a bit stiffer, then you see the wild-type cells are actually nicely, smoothly moving forward. But if you look at the wasp knockouts, they really have quite a problem under this agarose. So they can move, but they have kind of a stop and go pattern. So they make it a bit and then they stop again, they collapse, and then they move a little bit forward again. So they really have problems dealing with a stiff agarose, presumably because they can't deform it and cannot lift it up. So the conclusion here would be from these experiments that like the wave complex pushes the leading edge of the cell horizontally forward, whereas the wasp nucleated nucleated actin pushes up against the load that comes from the agarose, creates space and allows the cells to move forward. So the question is, how is the wasp then actually triggered? So how does it respond to the pressure? So Flo did a very straightforward experiment where he actually took a dendritic cell, looked at the actin, and then pushed into it with a micropipette. So if you push with a micropipette, there's a flush of actin polymerization in the cells. And if you look at the wasp knockout cells, they make way less of this response. So they really react with a burst of actin towards pushing into them. And we can quantify that, and this is really very much reduced without Wiskott-Aldrich syndrome protein. So the cells are also pushing back. So if you push with an atomic force cantilever into the cell and measure how much they push back, and you can actually see that the wasp knockout cells are softer. So the response, the actin burst response, really is a response of the cell to inward indentation, so to push outwards. So we think the cell has actually exactly this type of experience when it moves through a 3D collagen shell. So sometimes the cell, or very often the cell, has to move through a pore where it's constricted, and whenever it has to pass through this pore, there's a burst of actin here. And this leads to the dilation of the pore. we can quantify this actin burst here in the collagen gel, and we see that it's massively reduced in the wasp knockouts. And accordingly, when Flo let wild-type cells versus wasp knockout cells move in three-dimensional collagen gels, then they moved the same speed when the collagen gel was very low concentration. So when the pore size is high, they can do it nicely with and without wasp because they don't have to dilate a lot. But once you move to higher collagen concentrations, they need to dilate the pores and push away filaments, and the wasp knockouts get way slower than the wild-type cells. So this is how we envision it, but the question is now actually how is it really happening that the cell responds now to the geometry? So how can it sense the filaments, the collagen fibers pushing into it? So for this, Flo devised a more sophisticated setup where actually he built little nano ridges on surfaces. So these are, you can view them like collagen fibers on surfaces. So they are just with electron beam tomography built on silicon or glass surfaces. And we can vary them in size. But in the end, for the cells, they are just little indentations similar to the collagen fibers they are facing when they move forward. And then Flo pushed on the cells a bit with an agarose block. And if you look at the cells here that are kind of confined onto these nano ridges here, you can see that the wasp spots really localize to the nano ridges. And they strictly localize to the top of the ridges. So whenever something pushes into the cell, they react with localizing the wasp to the inward side of inward pushing. And accordingly to the localization of the wasp there is also a little burst of actin. So they also make the spots of actin. And if you knock out the wasp protein, they fail to make this actin accumulations on top of the nanoridges. So question is, how can they actually sense now that something is pushing into them? Because I have to say that these experiments here with the nanoridges, they were done with inert nanoridges. So they are chemically inert, or the cells are knocked out for the adhesion receptor so that they can't sense them chemically. So the only way they can do it is actually they can only sense the shape of the ridges. And one way we think this works, that they can recognize the shape, is really via curvature sensing proteins. And I think you all know curvature sensing proteins like F-bar domain proteins that sense inward curvature from the endocytic process. So they create inward curved membrane and at the same time can sense inward curved membrane. And when Flo used one of these F-bar domain proteins called CYP4 and labeled it with GFP and co-imaged it with actin, you can see the CYP4 is actually localizing to the same spots that then turn into actin spots. And if he puts the cells on the nanofibers, then he could see that they are co-localizing there with the actin spots. And via these experiments, you can also do space-time plots and see what's coming actually first. And what he saw is that WASP, of course, came before actin, because this is activating the ARP2/3, this is activating actin polymerization, and CYP4 is even longer before actin, and CYP4 comes before WASP. So they are sequentially recruited. So we really think that the way how the cell recognizes the extracellular matrix fiber is actually by sensing its own shape. So it senses the indentation of the membrane, thereby senses indirectly the shape of the fiber. It responds with the recruitment of WASP, which is known to be recruited by the F-bar domain protein, so by CYP4. And so WASP then activates ARP2/3. This polymerizes actin, and this is how a cell can then locally push away a collagen fiber. So this is now the picture how we think it works. So the cell assembles actin at the front to glide forward whenever there's free space by the wave complex. But whenever the cell is like confined locally by a fiber, then it senses the fiber and it locally pushes away the fiber by a burst of actin. This doesn't only work in fibrillar interstitia, but we also showed that for lymphocytes, actually in a packed environment of a lymph node, where it's a cellular environment, the cells also cannot respond to the compression without the WASP protein. And this is why the lymphocytes also migrate slower and cause all the adaptive symptoms of the Wiskott-Aldrich syndrome proteins. So to summarize this, we think that really cellular mechanosensing has not only the facet of that the cells adhere to something and test the mechanics by pulling on it, but they can also push into the substrate, and the mechanosensing is mainly, we think, done via sensing of the shape. But of course, that's early days here, and there are still a lot of things to find out in this area, and this is actually why we are always looking for new team members. And with this, I want to end and again acknowledge the people. So I think I mentioned most of them along the talk. So these are the alumni in the lab that contributed to this specific project I was introducing. This is the current lab, and here are some of the collaborators that contributed to the presented projects, and these are our funders. And before I want to end, I want to advertise something. So if you're interested in cell migration, there's really, in 2 months' time in Texas, in Galveston, there is the Gordon Conference for Directed Cell Migration. So that's really the top event in the field of cell migration. So there are not only people from immunology, but from all kinds of fields, lots of development people, cell biologists, neuro people, all interested in how cells actually migrate in a directional fashion. And there are still some slots free. So if you're interested, look at the webpage and register if you want to go. So with this, I want to thank you and the talk here.
Thanks so much, Michael, for a fascinating talk and a great journey through the basis of cellular locomotion. I just want to remind everyone that the next week speaker, Global IMMUNOTALK, will be presented by Maria Mittelbrum. And also that as always, the questions are going to be asked via Twitter. So you can search for the account Global IMMUNOTALKs. You can find the tweet that says ask questions here for Dr. Michael Sixt. And Michael will reply from the Twitter account from his lab mentioned here in this slide. So thanks again, Michael, and thanks everyone for attending.
Thank you.